Hi everybody, so today we're going to be talking about how a muscle works. So let's go ahead and jump right into this. And the first thing I'm going to do is, before we actually get into the muscle part, I'm going to actually describe what stimulates the nerve to contract. So we're going to look at the nervous system first and what happens. So this here is going to be a nerve that's come from the brain. Now remember, it's going to come from the brain, it's going to go down to the spinal cord, synapse with a nerve, and then it's going to go out to this area right here. So this is going to be my nerve right here. This round part that you see right here is going to be called the synaptic terminal. Okay, and basically the synaptic terminal, as you may know, is actually the end of our nerve, right? And the synaptic terminal is going to synapse with either another nerve, a gland, or in this case, it's going to be a muscle. This opening right in here is going to be called our synaptic cleft. Okay, and that's just the opening between the synaptic terminal and the receptors that are going to be on the other side. So that's going to be my synaptic cleft right there. And then if you notice inside of here, we have these blue things. So really quick, this is a cell membrane here, and then we have these here. These are called vesicles. I only have 3 drawn. You actually have a lot of these in here, but these are going to be called vesicles. So this is my vesicles right here. And the vesicles are going to contain something called a neurotransmitter. Now, neurotransmitters are responsible for carrying a nerve signal or impulse across the synaptic cleft. So again, this is my synaptic cleft right here. This is just the opening. Here's the next part of my muscle, right? My muscle cell. So we said this was a vesicle here, and the vesicle contains neurotransmitters like we said. The neurotransmitters are going to be these green things that are in here. And we call, in this case, the neurotransmitter that's going to make muscles move is called acetylcholine. So we're going to have acetylcholine in here. And the acetylcholine is going to come across this. But before we get there, let's take a look real quick. Again, this is my vesicle, and my impulse is going to come down the nerve. So the impulse comes down the nerve it's come all the way from the brain, it's gone in through the nerve, and now it's coming here to my synaptic terminal. When the impulse gets to the synaptic terminal, it is going to open up some calcium gates, right? So I'm going to have calcium, and calcium is going to come in. Now, attached to my vesicles, I have some proteins. And these proteins are going to be called snare proteins, okay? They're going to be called snare, S-N-A-R-E. Okay, they're going to be called snare proteins, and that's them right there. So what the calcium is going to do is the calcium is going to connect the snare proteins together. So again, I have snare proteins on my vesicle, I have snare proteins on my synaptic terminal membrane, and the calcium connects these together. I know that's kind of hard to see, but what this is going to do is, once those are connected together, it's going to pull this vesicle, and the vesicle is going to bond with the synaptic terminal membrane. So the vesicle membrane is the same thing as the synaptic terminal membrane. So these are going to bond. Those are going to bond. And now you can see we have an opening. So what's going to happen with that opening now is I am going to have my acetylcholine come out. So here comes my acetylcholine. Now, the abbreviation for acetylcholine is going to be ACH. So I just use ACH. On the opposite side, on my muscle fiber, or my muscle cell, I am going to have receptors. Okay, and on these folders, I'm going to draw a receptor right here. And this receptor is an ion gate, or an ion channel. When this acetylcholine comes, it's going to bond onto this ion channel. And when it bonds onto that ion channel, it's going to open up these gates here. And then what's going to happen is I am going to have sodium start going to the inside. Oops, that's not a +2, that's just a plus. And I'm going to have potassium come out. So sodium's going to go in and potassium's going to come out. Okay? When I get enough sodium on the inside, I'm going to change the inside of this enough that I'm going to get an action potential. And the action potential is going to start making its way along here. And I'm going to get to this in just a minute, but before I do, I want to talk about what happens here. After my sodium and potassium have gone through, I am going to have an enzyme that's going to break down this acetylcholine. The name of this enzyme is going to be called acetylcholine esterase. Remember, if it ends in ASE, it is a enzyme. So it's acetylcholine esterase. It breaks down my acetylcholine. Okay? So now, let's talk about what's going to happen here. When you look at this part that's right here, this is going to be called, or this is my muscle cell membrane, and it's called sarcolemma. So the sarcolemma is the muscle cell membrane. So this is my sarcolemma. And like I just said, it's a muscle, M stands for muscle, cell membrane. Okay, I don't have the bottom part drawn, but what's going to happen is this is going to come to my sarcolemma, and it's going to start traveling here. Now, if you notice, I have this big orange thing that's right here, and we call this a T-tubule. I have one here also. So this is called a T-tubule. And the T stands for transverse because it's going to go across this thing here. This whole structure that's right here is going to be called a myofibril. So this is my myofibril right here. That's the myofibril. Now, again, I'm talking about a muscle fiber here now, or muscle cell. So on the inside of my muscle cell, I have hundreds to thousands of myofibrils. Okay? When you see this blue part that's right here, this is going to be called my sarcoplasmic reticulum. Now, when you used to study cells, you might have recalled the endoplasmic reticulum. This is my sarcoplasmic reticulum. In a muscle, it's called the sarcoplasmic reticulum. So all this blue here is the sarcoplasmic reticulum. What this is right here is basically, these are tubes, so I can see my myofibril underneath this, okay? So this is my sarcoplasmic reticulum. Now, like we said, this is called a T-tubule. And if you notice, I have my sarcoplasmic reticulum. If you notice this part right here, we get like a bulge as this gets closer to the T-tubule. It's like the T-tubule tightens it, almost like putting your belt on too tight and having your stomach stick out. And I have another bulge that's right here, right? You can see we have one here and we have one basically right there. Where you see this bulging right here, these are going to be called terminal cisterna. Okay, so this is going to be my terminal cisterna. Now, some books will call this the terminal cistern. Some books call it the terminal cisterna. So it's going to be right here, and this would be one here, right? If it's plural, we call it terminal cisternae. Okay, if it's plural. And I might as well put this right there. So there's another one. So there's my terminal cisternae right there. So if I take one T-tubule and my two terminal cisterna, right? One T-tubule and two terminal cisterna, one T-tubule plus two terminal cisterna, or in this case it'd be cisternae, right? That's going to make up something called a triad. That's an equal sign, by the way. That's going to make up something called a triad. Okay, so my one T-tubule and my two terminal cisternae make up a triad. Okay? So now, here comes my impulse. Remember, we're talking about the impulse, right? Here comes my impulse, and it's going to start making its way down into the T-tubule. Now, I'm just showing it here, but it would continue, and it would go here, and there's a whole bunch of these in there. Inside the T-tubule, I have something that's called tubule proteins. So I'm going to draw a tubule protein here, and I'll draw another one here. And these are going to be called my tubule proteins. So this is tubule proteins. And my impulse now is going to come down through here, and it's going to basically activate these tubule proteins. And when it does, it's going to change their shape. Now, Before I get to that, on the inside of my sarcoplasmic reticulum, I have calcium. Okay, so there's calcium all in here. Now, if you recall, we just got done talking about the role that calcium plays over here in the synaptic terminal, right? But now we have calcium again. So when this impulse comes down, it's going to change the shape of these tubule proteins, okay? Now in real life, these are not square, all right? And I'm gonna use the symbol for change to show a change in shape in my tubule proteins. When I get the change in shape in tubule proteins, what that's going to do is that is going to open up some calcium gates, right? That are in here. So I'm gonna open these calcium gates here, and now calcium is going to start coming out. So I'm making it look like this calcium is actually coming out of my myofibril, but it's not. You are looking at the outside of the myofibril here. This is actually going to leak to the inside. So let me show you what I'm talking about. Imagine I have this here and I pull this off and now I'm going to turn it towards you, and now you're looking at the inside of the myofibril. My fingers would be where the sarcoplasmic reticulum is, and inside would be my myofibril. So if I were to draw it, I'm going to draw it like this, like the tube, right? And then I'm going to draw my sarcoplasmic reticulum around it. Okay, and just once again, this is my myofibril. This blue part would be the sarcoplasmic reticulum. I'm just going to write SR for sarcoplasmic reticulum. In fact, just so you remember, I'll put SR right there. So there's my sarcoplasmic reticulum. This would be it. So I have my calcium. So like I said, imagine this is my calcium. I have my tubule proteins. I'm just going to show these as if they've already changed shape, right? And then the calcium is actually going to go from the sarcoplasmic reticulum into the inside. So now I'm going to get calcium on the inside here. Okay. To give you another idea of how this works, imagine my paper towel roll right here. The white part here would be my sarcoplasmic reticulum, and then you're looking at the inside. Let me see if I can get a little bit more white area in there. Okay, and so we have the myofibril. This is the inside of the myofibril. The tube would actually be the myofibril, and then the calcium is going to go from here to inside here, is the way that this is happening, okay? So let's go on and look at the inside of this now, right? So now my calcium is drained or has rushed into the center of this, okay? Inside of here, we have something called the sarcomere, which we're going to look at in just a minute. But I want you to remember this calcium because I'm going to be talking about it in just a minute. So let's take a look over here. And right there, what you're looking at now is called a sarcomere. Okay, so on the sarcomeres basically go, this is called the Z line, or some books call it a Z disc. This is a Z disc. So my sarcomeres basically go from one Z line or Z disc all the way over to the other Z line. That's going to be the sarcomere. The sarcomere is going to be my functional unit of a muscle contraction, okay? So, or of a muscle. So let's go ahead and take a look at this. If you notice, we have a few things here. I have this here. You have two types of myofibrils. So let's go like this. You have two types of myofibrils, and they are called actin and they're called myosin. So you can see this is a myofibril, this is a myofibril, this is a myofibril. This is my myosin here. This is the actin, this is the myosin. So this would be my actin, and we just call it actin filament. Okay, so the actin, I'm saying myofibril, I mean these are filaments. You have two types of myofilaments. It's actin and it's myosin. This is the actin, this is the myosin. So my actin is the thin filament. Okay, and I always remember this because we say it acts thin, right? So it's a thin filament. My myosin is the thick filament. So this is my myosin here. And this is the thick filament. Okay, so I have a thin filament and I have a thick filament. Okay, and those are both myofilaments, not myofibrils, myofilaments. Okay, but just alone, we just call them the thin filament and the thick filament. So, if you notice here, this is the way the books usually draw them. It's just a straight line, and then they're going to show what I'm talking about now. This is basically, we're going to take a look inside this here. We're going to take a look inside this here, and it would look something like this here. The way that actin actually looks is it looks like two strands of beads all wrapped around each other. Okay, so let's draw this right over here. And let's see, I'll go green and blue. Where's my red? Okay, so if we go like this, this is going to be one part of my actin. Okay, like we said, it's like two strands of beads that are around each other. And then I'm going to draw this here. Let me find my red real quick. There's a red. So then if I draw this here, this is going to be my other part of actin. Like I said, it's like two strands of beads that are wrapped around each other. Okay, so that's the actin. So all I did is I took this here and I drew it up here. Now, inside of these, we have something called active sites, which some books say they're depressions, some books just say they're a little hole. So let's take a look at these. And I'm just going to make these active sites. I'm going to make it look like all of these are actually facing out towards us. But in actuality, they're going to spin all the way around this actin. Okay. Then on top of that, we have something that's going to, well, before we even get there, let's go like this. We're going to call this the active site right here. That's an example of an active site, and that's an example of an active site. Now, let's say I were to take one of these little active, or one of these, one piece of this actin off, right? And I was to draw it. So to give you an idea of what this would look like, I'm going to take this off, and now we're going to draw it. So I'm going to draw it like this, all right? And your actin would look almost something like this here. Okay, so that's going to be my piece of actin right there. And then this part down in here is going to be my active site. Okay, so when we talk about actin, what we want to know is that the actin is like two strands of beads wrapped around each other, and they have an active site. But there's some more things to go over real quick. These active sites are covered a lot of the time. Okay, so when your muscles are not contracted, these are going to be covered. So I'm going to use the blue to demonstrate that. They are going to be covered by a protein, and the name of this protein is going to be tropomyosin. So I'm going to go like this, and then I have tropomyosin. And the tropomyosin is actually the protein that covers active sites. So it covers active sites. And we're going to go into what active sites do in just a minute. But to give you an idea, let's move this back just a little bit. To give you an idea, well, before we get there, I want to go through one more thing. There's another protein that's in here, and we're going to call this protein troponin. So I'm going to make troponin look like a bunch of little tacks. And what troponin does is troponin, let's go one more right here, and this way I can go like this. Troponin holds tropomyosin in place. So troponin holds the tropomyosin in place, okay? So now, let's take a look at what's going to happen here. This here, pretend like this is my actin here. This is going to be my active sites, right? This is my actin. These are active sites. This is my tropomyosin. So my tropomyosin is going to hold, or it's going to cover the active sites. My fingers would be the troponin, right? So the troponin is holding the tropomyosin in place, okay? So now let's take a look at what's going to happen. If you remember, we were talking about calcium earlier. So when the calcium came down into the inside of the myofibril, right, it basically bonds with this troponin. It's going to bond with this troponin, and when it does, it changes the shape of the troponin. So now I'm just going to change the shape here. I'm going to change the shape a little bit, right? And let's go like this. So let's say it's changed the shape, right? Change the shape, change the shape. Now when it changes the shape, it no longer holds the tropomyosin in place. The tropomyosin is then going to move, and when the tropomyosin moves, now we can see the active sites. Okay, so it's going to look something like that. So let's look at this again. So now there's my troponin. There's my actin. Here's my tropomyosin. There's my active sites. This is covering this, right? It's covering this. My fingers would be the troponin. Now the calcium has bonded with my fingers, changed their shape, and now my active sites are exposed, right? The tropomyosin was out of the way, and now my active sites are exposed. So let's look at what's going to happen next. This here is my myosin, right? The blue line is my myosin. And you can see we have pieces that are sticking out here. So let's take a look at myosin. And my myosin is going to look like this. My myosin is going to have a tail. Then it's going to have something called a hinge. Then it's going to have a head, but the head is actually made up of two different parts. So one head is going to be for active site attachment. And the other head is going to be for ATP attachment. Okay, so I'm going to have two different heads on there. So now, this is what's going to happen now. When we go to move the muscle, right, when we want the muscles to contract, I'm going to draw my actin again here, just my one strand of actin, right? This is going to go up into the active site. So this will go up into the active site, right? Here's my active site. Here's my actin, and this is going to go up into here. Okay, you can see I've done it right here. This is actually a demonstration of what's going to happen. Okay, so it's going to go up into there. Now, when it goes into there, this is going to pivot. It's going to pivot, right? So right here, that goes up into here, and this is called a cross bridge. We'll talk about this in a minute. And then it's going to pivot, so you can see it's bent there, right? And when it pivots now, it moves this actin this way. It's going to move the actin that way. So it goes up in there and it pivots and it moves the actin that way. Now this is happening all over here. Okay, so even though I'm just showing it right here, it's actually happening all over here. And the thing is, I'm showing this in 3 different steps, but in actuality, all of these would actually be doing this at the same time. So this is like my step number 1, this is my step number 2, this is after it pivots, and then it releases, and then what it does is it finds another active site to go into and do it again. Okay, so it goes into the active site and pivots, right? And this is where it's pivoted, and it moves the actin that way, and then it comes out, and then we're going to talk about what happens in just a little bit, but then it's going to go back into another active site and do this again to push it that way. On this side, it's the same thing. Now I'm showing step 1 and step 2. This is step 1, and this is step 2. So it's going to go in and pivot, and it's going to cause this one to move that way. So I got both these now, if these are pivoting, right? If they're pivoting, they're going to move the actin this way. And as they move the actin this way, my muscles start to contract. Okay, so this goes in and pivots, this goes in and pivots, and it moves them this way. So let's take a look at this again. This is, once again, are my active sites. This is my actin. This is my tropomyosin. Right, there's my tropomyosin. And then what's going to happen is, like we said, my calcium is going to bond with the troponin, which would be my fingers. The troponin is going to change shape, and when it does, it allows the tropomyosin to move, which is going to expose the active sites. okay, to expose the active site. So now, this is going to be my head that's going to go in here. So let's say this actin is this one right here, and I want it to move that way. What would happen is if this is my myosin and this is the head, this is going to go into an active site, like we said. Let me go like this. Going to go into an active site and pivot. When it pivots, it moves the actin forward. Then it comes out and it just finds another one. It doesn't have to be the next one. It's just going to find another one. And then it's going to pivot again, and it's going to continue to move the actin that way. On this side, it'd be the opposite, right? This is going to go in to the myosin head is going to go into the active site and pivot. And now it moves the actin this way. And as they come closer together, like I said, as they go towards each other, the muscle contracts, right? So that's going to be what happens in a muscle contraction. Some things I want to go over real quick is this. When we have the head of the myosin in the active site, we call this a cross bridge, right? A cross bridge formation. Okay, so a cross bridge is when the head is in the active site, okay? The fact that we said this was actin, right? All the red lines were actin, and actin is a filament, and myosin is the blue lines, and myosin is a filament. As the actin moves, it's sliding across the top, or it's sliding along the myosin, right? So my actin's sliding along the myosin, so we call this the sliding filament theory. So the fact that my actin slides along the myosin is the sliding filament theory. Okay. One other thing I want to talk about is, if you notice, I make it look like I have one myosin and then I have two actin. Okay, so there's one myosin and then I have two actin. Technically, for every myosin you have 6 actin. This is a tube, right? It's round. So this is going to look like this. I'm going to go like this. There's my myosin. This is as if you're looking at this now this way. So I took this out and I turned it this way, okay? Or I'm looking down like this. So there's my myosin, and then I would have 6 actin going around it. Okay? So to make my cross bridges now, imagine I got 3, 4, 5, 6, yeah. My cross bridges now would look something like this. And then all of these would contract at the same time to bring the myosin, to move the myosin, okay? So we call it the sliding filament theory. When these are together, it's a cross bridge or cross bridge formation. One last thing that I want to talk about is the ATP attachment that we're talking about a minute ago. So we said that this side here is for ATP attachment, right? This is for ATP attachment. So let's take a look at this real quick. And this is what I'm going to do is, let's say I have my myosin like this, there's the hinge, and then I got the head here, right? And then I have my ATP attachment part. So now my head is inside of the active site, right? So there it is inside the active site. This is my cross bridge formation here, right? And then I have something attached to this. Now I'm going to draw this real big, but it's really not that big. I just want you to be able to read it. I'm going to have something called ADP plus a phosphate, and it's going to be attached to this part of the head that's called the AP, is responsible for ATP attachment. Now, if you notice, that says ADP plus PI. So we're going to get to this in just a minute. What we said happens next is this is going to move. It pivots. So it's going to pivot this way. So I'm going to just draw a little line like this. Now, my myosin has pivoted. Okay, so there's my myosin. There's my actin. Okay, it's pivoted, and because it pivoted, it moved my actin that way. What happens when this pivots is this ADP and the phosphate, it leaves. Okay, so I'm going to draw this right here. There's my ATP plus the phosphate. And if you don't understand these, I'm going to explain these in just a minute too, right? So then what's going to happen is this is going to go to the mitochondria, and my ADP, it's going to attach this phosphate back onto this. So it's going to attach this phosphate back onto this. So I got ADP, phosphate attaches back onto this. This becomes ATP. Okay, that's going to happen in the mitochondria. If you recall when you're studying cells when you were younger, the powerhouse of the cell is ATP, right? That's basically what we've made now. So now, if you notice, I don't have ADP attached there. So what's going to happen now is we are going to have this like this, and then this ATP is going to attach onto this here, okay? So this ATP is going to attach onto this part that's right here. So I'm going to draw ATP, and when it does, it releases energy. So this is going to release energy now, okay? And when it releases energy, it's going to cause this to go back to the position it was in. So right now, if I can get the angle on this, right now it's basically going this way. It's going to come back this way, so it looks like this. Okay, so this now comes back, and it looks like this. However, because this used energy, I'm going to explain this again in just a minute, so be patient for a second if you don't understand this yet. This is now going to become ADP again, plus a phosphate, okay? Now it's ready to go. Now it's ready to go. So now I can go into another active site and then do that again. And this continues, right? That's why I have these going back and forth, because this will go from ADP with a phosphate, then it gets attached, then it becomes detached. So it goes back and forth. So let's take a look at this right here real fast. Let me go over here. So now, here's what's going to happen. Is we have a molecule, let me see if I can go over just a little bit more. You have a molecule, right? Like we said, we had ADP, and ADP is made up of adenosine, all right? There's adenosine, and then we have a phosphate attached, and it has another phosphate attached, okay? And then it has another phosphate attached. So what we say is it's adenosine, and how many phosphates do we have? 1, 2, 3. What's a prefix for 3? Tri. So we have adenosine triphosphate. But instead of saying adenosine triphosphate all the time, we just go A, T for tri, and then P. So this is equal to ATP. Now, this bond right here between these last two is a weak bond. So here's what's going to happen when I need energy. When I need energy, what's going to happen, again, this is my adenosine, right? There's my phosphate, there's a phosphate. But when we have a weak bond here, what's going to happen when we need energy, this bond breaks. And when it breaks, we get, well, actually, I don't want to draw it right there and confuse people, so I'm just going to go like this. Okay, so I have this bond here, right? Which I basically just drawn an ATP, right? But I want to concentrate on this bond right here. So when I need energy, this bond breaks. Okay, it's the same as that bond there. I'm just drawing it here. Okay, and then when that happens, energy is released. And I can do things such as put that myosin head back in position. But again, this is no longer attached to there, so I'm just going to say this is there. Now this becomes an inorganic molecule. Okay, so now what happens is now I have ADP. I have two phosphates, adenosine diphosphate, ADP, plus with the phosphate there. So this becomes ADP. And then like I said, then it goes to the mitochondria, and the mitochondria attaches this back on, and now it's ATP. Then when I need energy, this breaks off, and it becomes ADP. Then it goes back to the mitochondria. So it's a cycle that's going to go back and forth between these two. Okay? So that's a cycle. So anyways, that's it for muscle contraction. I hope you enjoyed the video. It was a lot, okay? But I hope you enjoyed the video. If you liked the video, please hit the like and subscribe button, and we will catch you next time. Thank you so much for watching.